Hello, everyone. I'm Dr. Arpita. The topic for today's discussion is vaccines. This is a very popular topic as we all have come across this word at some point of our life. Hmm? So, how do we define the term vaccine? Vaccine can be defined as the preparation of bacterial, viral, or other pathogenic agents or of their isolated antigens, which is administered with the objective of stimulating a patient's protective immunity. So, in a very simple way, we can define vaccine as something that helps us to fight against a disease, or it plays a very important protective role in our body. Okay, so vaccine, see, this word vaccine owes its origin to the Latin word vacca, vacca, meaning cow. To be very precise, Edward Jenner did not introduce the term vaccine. It was Pasteur who did that. So remember, the term was introduced to us by Pasteur. All right? Yes. See, after primary exposure of antigen to immunocompetent lymphocytes, there occurs an initial but slightly delayed immune response, which is known as primary immune response. Now, this primary immune response peaks on approximately the 14th day of antigen exposure. Remember this, it peaks on the 14th day of antigen exposure. Okay. There are two main outcomes of this primary response. The first being uh, the specific immunocompetent cells, the B and T cells are activated. And the second one is what? The more important one, rather, the memory cells are formed. So what happens? Here you see, first comes your primary immune response. Okay. So what did I say? This primary immune response peaks on approximately the 14th day of antigen exposure. Fine. And the two main outcomes are what? The first and the second one, which is the more important one. First is B and T cells are activated. Sorry. T cell activation. And second is what? Memory cell formation. Fine. The secondary response usually occurs within 2 to 3 days. Well, it must also be mentioned that a primary immune response elicits the formation of IgM antibodies, while a secondary response uh, protects the host with higher affinity IgG antibodies. So you need to remember two things. Primary immune response elicits formation of IgM. This one, it elicits formation of IgM, whereas secondary response protects the host with higher affinity IgG antibodies. Okay, it is the rapidity with which secondary response occurs upon secondary exposure to antigen that protects the host against any potential threat by repeated exposure of pathogen. This is something to be kept in mind if you are really planning to study vaccines very well. Thus, vaccine is basically an antigen or its component that can induce secondary or adaptive immunity in the host. Clear? Okay. So now we'll uh, talk about vaccine and its types. This is where questions come from. You need to know what are the various types of vaccines available. Okay. So first we'll start with natural live vaccines. Okay. We'll first start our discussion with natural live vaccines. Okay, there are several types of vaccines which are conventionally used. They are natural live vaccines, then inactivated vaccines, live attenuated vaccine, toxoids. Many are there. We'll discuss each one. Say, coming to natural live vaccines. Okay. See, these preparations include natural, non-pathogenic organisms. Okay. But remember, natural live vaccines could turn pathogenic in a host. Currently, live natural vaccines are not in regular use. They are rarely used. Apart from the historical cowpox virus vaccine, no other natural organism are usually used. So, if we are quoting an example here, which is the cowpox virus that I'm talking about, cowpox virus used for immunization against smallpox, right? It has historic importance, but now it has become redundant. Okay. Apart from this, simian rotavirus, this is used in vaccinations against gastroenteritis. Hmm. This rotavirus, it belongs to the rotaviridae family of viruses, and this virus causes gastroenteritis among children, which manifests as severe diarrhea, vomiting, and is also responsible for about more than 5 lakh deaths per annum in developing countries. Fine. Our next one is our next topic is inactivated or killed. Inactivated or killed vaccine. What happens here? See, some live pathogens were there. Suppose these are live pathogens. Then these live pathogens are subjected to inactivation, and these remain no longer active. Okay. Now, this killed pathogen becomes your vaccine. Okay. Now, here the disease causing germ is killed using what? Either heat or some chemicals or radiation, or this inactivation is done using what? Heat, radiation, chemicals. All these are used to kill the pathogen, right? This was live pathogen. This was your live pathogen. Fine. And these, mind it, these inactivated or killed ones are stable, easy to store and transport. Remember this point. These are easy to store and transport. They are safer and stable. Safer and stable, right? See, there are various advantages apart from these, no mutation or reversion to wild type forms is possible. It's not possible whatsoever. Why, because it is already dead. How can mutation occur in this? So no mutation or reversion to wild type forms possible. This is another advantage. No mutation, reversion to wild type uh, forms since they are dead, right? And it provides sufficient humoral immunity, right? And then they are heat stable, as I've just told you. And it can be safely used in immunodeficient patients. It can be very well safely used in immunodeficient patients. So the last point, if you write, used in immunodeficient patients. Clear? Okay. Now, there are few disadvantages too. Everything around us has some amount of advantages, some disadvantages. Nothing is fully good, nothing is fully bad. Fine. So these also have certain disadvantages. Yeah, one more advantage I didn't mention, it doesn't require any cold chain for it. We'll talk about this later. When we'll talk about live attenuated ones, we'll talk about the requirement of cold chain and all, but here it's not required. Fine. Now see, now let's talk a little about its disadvantages. The disadvantage is that they stimulate weak response and generally requires booster doses. Fine. So see, if we enumerate the disadvantages, it will be what? Weak cell-mediated responses, weak cell-mediated responses. Second, it requires booster doses. Right? Why do you think it requires booster stimulation? Because again, it is dead. It cannot replicate inside the host. Therefore, you need to administer this from outside to the patient. Because in the patient's body, this is in dead condition. It cannot replicate. Clear? Then this is more costly. And the last but not the least, inadequate killing of virulent organism can result in the occurrence of full-blown disease. Very, very important. Inadequate killing. Inadequate killing can result into occurrence of the disease. Occurrence of the disease. So these are the few disadvantages that you should know. You cannot miss out these disadvantages. Right? Now, apart from the advantage and disadvantage, we need to know some of the very, very important examples of these inactivated or killed vaccines. Fine. Now see, two types are there. Killed viruses can be used as vaccines. Killed bacteria can be used as vaccines. Fine. Now we'll talk about some examples. Pertussis vaccine, then typhoid, cholera, typhus fever, okay? These vaccines are made from killed or inactivated bacteria. Now coming to viruses, polio. Polio vaccines, you have heard of it, right? Rabies, then influenza. All these you have already heard. Now see, let's talk a little about this polio one. It's very interesting. Why, you know? Why this is so important, you see. This ink is not showing up. Yeah. We'll use this. See, uh, this is something very important. You must remember this. One is inactivated or killed. The other is live attenuated. Fine. Now see, in this inactivated one, hmm, we'll write the example now. When we'll do live attenuated, we'll talk about that also. The Salk polio. The Salk vaccine, the Salk polio vaccine falls under the inactivated or killed one. It is used earlier and injected intramuscularly. Remember this, used earlier and injected intramuscularly. Fine. And the live attenuated one is the Sabin vaccine. The Sabin vaccine is the one which is presently used. Oral dose. Mr. Bachchan comes up with this advertisement, no? The promotion of this, Do Boon Zindagi Ki. This one is that, is the Sabin vaccine. So next time when you see him talking about Do Boon Zindagi Ki, events are, if I'm not wrong, Mr. Sachin Tendulkar, Mr. Amitabh Bachchan, these people all come up with this promotion to make the mass aware about this. So Sabin vaccine is the one which is presently used, the oral dose one. It's a trivalent preparation, poliovirus type 1, 2, and 3. Hmm. So let's write that also, trivalent preparation, poliovirus type 1, 2, and 3. Poliovirus type 1, 2, 3. Fine. All right. So now next we'll move on to live attenuated vaccine. Okay, our next topic of discussion is third is right. It's a third. Hmm. Live attenuated vaccine. Let's see what happens here. See, in live attenuated ones, what is happening? The live pathogen, as I had drawn over there, suppose these are the live pathogens, okay? Now, this live pathogen is attenuated. Attenuated means what? Attenuated meaning is what? Weakened. It is weakened. Weakening is done. Okay. They are not killed. They are just made weak. They are weakened and results into, I'll just shade it to make it look little different. Attenuated ones, attenuated non-pathogenic form. This is the attenuated non-pathogenic form. Clear? Now, what happens here? It is made by, it is made up of living organism, but it's made weak. Hmm. It has been weakened by mixing it with different concentrations of formaldehyde. They stimulate excellent lifelong response. Remember this, they stimulate excellent lifelong response. Fine. You have heard of the yellow fever virus? The yellow fever virus is attenuated by passage through mice and chicken embryo cells. While the Sabin one, this ye wala, Sabin vaccine, which I was just talking about, the Sabin type 1 poliovirus is attenuated by growing the virus for prolonged periods in the epithelial cells of kidneys in monkeys. So there are different processes to achieve this. Okay. See, microorganisms are attenuated or weakened so that they do not cause any disease. This is a very important point. Why are they weakened? They are made weak. Why? So that they do not cause the disease. So that they do not cause the disease. Clear? Attenuation can be achieved by growing the pathogenic microorganism for a long period in a foreign host, such as embryonated eggs or tissue cultured cells. This is one way, and the second and more famous attenuation was successfully carried out by, remember these names, two scientists, Albert Kalman and Camille Guerin. You will understand why am I talking about these two people. Their name comes in the context of BCG. You have heard of this vaccine, BCG? Children are administered with BCG doses. Kamat and Guerin grew the bovine strain of Mycobacterium tuberculosis for several years on a medium containing increasing concentration of bile. Okay. Now, this in vitro cell culture of pathogenic organism hmm, changed it into a less virulent or more suitable form of bacilli known as BCG, Bacillus calmette-guerin. In this context, you must know the term BCG. Here I'm writing it a little big. BCG. You just cannot forget this. Bacillus calmette-guerin, BCG vaccine. Okay. It's a commonly used vaccine against tuberculosis. The commonly used vaccine against tuberculosis. Attenuated viruses are also used as vaccine. Okay. The vaccines of polio, yellow fever, and measles viruses being the most successful ones. Hmm. They stimulate excellent response and it's lifelong. Remember one more important thing. If they are not properly attenuated, that means what? They are very strong, ready to produce disease. What will happen then? If not properly attenuated, then there is a chance of full-grown disease in the body. Now see the misfortune hidden here. You're administering the vaccine for the good of the patient, but unknowingly You are sending the pathogen to the body of the patient, I mean, to the body of the person who is soon going to be a patient, right? Apart from this BCG, we also have the oral polio vaccine, the MMR. Heard of this? You must have heard. Hmm? MMR means measles, mumps, rubella. MMR, M stands for measles, mumps, rubella. MMR. Okay. Then these yellow fever, rotavirus, all these I was just talking about. All these are important examples of live attenuated virus. Then our next example, so our next name on the list is toxoid. Heard of this before? There is no one who has never heard of toxoids. These are used against bacteria that secrete harmful toxins. The toxin in an inactivated form is used to prepare toxoids. Okay, now these toxoid vaccines are these diphtheria vaccines, tetanus vaccine, all these are toxoid, right? Okay. Actually, why this came into existence is we have seen that the virulence of some pathogenic bacteria depends primarily on the production of exotoxin. Now, what are exotoxin? The question comes in. See, every point, every topic should be very clear. Your idea, your concept, everything should be very clear. This is not about just, mugging up. This is not at all about just, sitting and mugging up everything. This is about learning something new, understanding the concept so that you can build in your own concept, right? See, exotoxins are toxins. These are toxic substances which are secreted by living cells. Fine. They are highly potent and elicit major damage to the host. Now, these are extremely heat labile. Remember this point. These are extremely heat labile. Correct. Can you name a very common toxin producing bacteria? Can anybody name a very common one? Yes. Clostridium botulinum and Pseudomonas aeruginosa. These two are very popular toxin producing bacteria, right? Okay, now let's talk a little about this. Have you heard of the word tetanus? You must have, right? Some bacterial pathogens such as diphtheria and tetanus bacilli produce exotoxins that induce several characteristic symptoms associated with these infections. Or in simpler word, we can say the harmful part of these bacteria is their exotoxin. So, so now the point is that if these exotoxins can be neutralized means if this can be made inactive, non-toxic, hmm, inactive most importantly, then what might happen? What will happen? Yes, any idea what might happen? Then the disease will not occur. As simple as that, the disease will not occur. isn't it? Now, these exotoxins are isolated. Should write this. What is done here? See, exotoxins are isolated and chemically modified. Okay, they are isolated and chemically modified. Clear? How is it modified? Usually using formaldehyde so that their toxicity is lost. I'll change the color of the ink so that you remember. Their toxicity is lost, but the immunogenicity, remember, immunogenicity is not lost. Remember this line for toxoids forever. I said their toxicity is lost, but not their immunogenicity. If their immunogenicity is lost, then what is the use of using this as vaccines? It's just useless, right? These non-toxic yet immunogenic derivatives of exotoxins or toxoids are commonly used in vaccines. Okay. Now, vaccination with toxoid induces anti-toxoid antibodies, which are capable of binding the toxins and neutralizing it. So, basically, what we can say, toxoids are modified toxins that have lost their toxicity. You can say this, no? Toxoids are modified toxins that have lost their toxicity, but not immunogenicity. They are either generated by chemical treatment, that means using formaldehyde, or by heat treatment. As I said, they are heat labile. Okay, let's talk about some advantage of using this. They don't cause the disease. They are safe and stable. They don't cause the disease. Safe. They are stable. What disadvantage does it have? It requires, what does it require? It requires booster dose. All right, they require booster dose. May also require some adjuvants. Now, coming to examples. Examples are what? You know this, I suppose. Hmm? Tetanus vaccine, diphtheria vaccine, all these. Huh? Diphtheria vaccines, all these are very common examples. Clear? Great. Now we'll come to the next one. Now we'll come to the fifth. Yeah, the next one is subunit vaccines. It uses only the antigen that stimulate the immune system. Uses only the antigen that stimulates the immune system. Clear? Like, like the capsular polysaccharides. Well, if I say just one example, production of hepatitis B vaccine. Very, very important point. Production of hepatitis B vaccine. Okay. See, X gene codes for the capsomere protein. We write, we'll understand better. Say X gene. It codes for the capsomere protein. Right? The capsomere is digested with the help of protease to get the X gene of DNA of the virus. Hmm. Now, the segment of the DNA is cut with restriction endonuclease to isolate the X gene. Follow each line that I say. The, what did I say? The segment of the DNA is cut with restriction endonuclease to isolate the X gene. So, restriction endonucleases are used to isolate the X gene. Okay. This X gene is the one which is going to be loaded onto a plasmid vector. This is loaded onto a plasmid vector, right? That same restriction endonuclease, which was used for cutting the viral DNA, that same RE is used to cut the plasmid also in order to generate similar cut ends, or else what will happen? The ligation process will not be possible. Hmm. The vector is then inserted into the bacteria, the bacterial cell, which starts the expression of the X gene, and then what does it do? And then it produces the required protein. Fine. Now, this protein is then used for vaccination. All right. Now, after loading this one onto the plasmid vector, hmm, as I told you, the same restriction enzymes are used to cut the plasmid so that similar cut ends are generated, or else ligation later will not be possible. The vector is then inserted. Vector is then inserted into a bacterial cell. The vector is then inserted into a bacterial cell. It starts the expression it starts the expression of the X gene. And and what does it do? And produces the required protein, and this protein is then used for vaccination. Protein is then used for vaccination. Okay, now let's talk about examples. The pneumonia, meningitis vaccines, these are bacterial ones, and virus example I have already told you. From viruses, hepatitis B, and from bacteria, meningitis, then pneumonia. Hmm. In this meningitis and pneumonia, the purified polysaccharide is used. Then, now coming to DNA vaccines, this is also a very interesting one. What happens here? See, this isn't nice. In DNA vaccines, what happens? It uses the genetic material of the pathogen directly to be used as vaccine. Hmm. In DNA vaccines, what is actually happening, you see, the genetic material is directly being used. Okay. The genetic material here is directly being used as vaccine. Remember one thing. The DNA is not used as the antigen, right? The DNA is not used as antigen. Rather, what does it do? It codes for the antigen. It codes for the antigen that is expressed inside the host cell or tissue. Okay. Actually, the term DNA vaccine is a misnomer. Huh? It wrongly implies that DNA is used as an antigen, but actually it is not. This is not the case. Hmm. So here, see, the gene of interest is taken from the pathogen genome. It is taken from the pathogen genome and introduced in the host through recombinant vector. A recombinant vector is used here for insertion. See, DNA vaccines are very stable and can be stored at room temperature in dried form or in solution. Okay, this is one example. Sorry, advantage. Very stable, extremely stable. Can be stored at room temperature in dried form. Okay, or in solution, dried form, or in solution, anything. Now, see, conventional vaccines have to be stored in special refrigerators called cold chain. Here, this cold chain and all those things is like not required. You can store it in room temperature. See, muscle cells, they take up DNA more easily than any other cell and hence are used as target cells in DNA vaccines. The muscle cells are used as target cells. For these type of vaccine, what is the target cell? Target cells for DNA vaccine is what? Muscle cells, right? Clear. Now we have almost come to the end of the list. Next is recombinant live vaccine. Recombinant live vaccine. Well. Before we start this, we didn't write the examples of this, no? Examples of DNA vaccine is, say, rabies, influenza. All these. Hmm, fine. So this was our next one in the list. Recombinant live vaccine. Recombinant live vaccine. Here what happens? See, the procedure is same as that of hepatitis B vaccine, which I was just discussing. The process is same till the separation of X gene. Here we had talked about separation of X gene, remember? Till the isolation, the process is same. Now this X gene, the isolated X gene is then ligated to vaccinia virus. The isolated X gene is 1 second. Ligated to vaccinia virus. Huh? It is ligated to vaccinia virus genome. Thus, recombinant vaccinia genome is created. Thus, recombinant vaccinia genome is created. Vaccinia genome is created. Clear? Now, fresh virus is generated by in vitro packaging of the genome that carries the gene of interest. Now, gene X will be expressed and our body will form plasma cells followed by memory cells. This is what happens. And the last in our list today is cassette vaccines, the last one to be discussed. Okay. This is also an interesting one. What happens here in cassette vaccines? Vaccinia virus genome is used as a vector for carrying gene of interest from one or more of the pathogen, right? You have heard of this cassette vector or, cassette vaccine. Yeah, one more. Yeah, one second. Yeah. This here also, the vaccinia virus genome is used as a vector. It carries the gene of interest from more than one pathogen. Fine. Another thing, before we end, we must discuss one term, that is herd immunity. Out of it, herd. Herd immunity. What is that? See, in a large group of individuals, if the majority of the individuals are immunized, what will happen? Then the pathogen will fail to infect. That is, cause the disease in non-immunized persons also. So the non-immunized people get the advantage of staying together with immunized people. This is called herd immunity. Fine. So this is how we come to the end of our lecture. And as I generally do, before ending the lecture, I'll ask you a question. Now let's see who can answer this. Which of the following vaccine will not pose any problem in immunocompromised person? I'll give you four options here. You'll have to tell the right answer in just few seconds. Option A, measles. Option B, mumps. Option C, BCG. And option D, pneumococcus. Yes, the last one is the correct answer. So this is how we come to the end of our lecture. I hope you have enjoyed learning about vaccine. If you have any confusion whatsoever, please feel free to get back to me in the comment section. Till then, thank you and goodbye.